Hey guys, welcome back to Computational Biology with Python. In this video, we'll be doing more with the Biopython suite of tools. Last video, we did a little bit of pulling out records from a sequence file that had multiple organisms for a single gene. And we'll now go into extracting data from these sequence files. So let's go ahead and jump right on in. All right, so if you're just loading up, you'll probably have to import seq.io again. Let's just run that real quick. Okay, so kind of a refresher. Record, iterator equals seq.io parse. We're going to use that ls orchid gbk, and then genbank. Okay, so instead of above the last video, we did use the gbk and not the FASTA file. I couldn't remember. Okay, so we're gonna load that. Remember, this guy has all of our different organisms and all of our different sequences. And then first record equals next for record iterator. So that's our first record. Remember, next starts at the beginning and goes through each one. And then we're going to print the first record. Record. All right. Here we go. So we got C. europeanum. This is the ribosomal RNA gene. You got the molecular type or molecule type DNA, linear topology, etc. So this is what this is doing. When we say print, it gives us kind of a human readable version. So, it breaks it up nice where everything's on a new line. It gives us not the whole sequence, but the last 3 nucleotides and the first, whatever this many is, 30, I guess. And some of what the references are, things like that. We can also print first record annotations. So you can see these are all the things that are in the annotation list. You have the molecule type. This is really just taking this stuff right here. You can see my cursor right here and printing it directly without the rest of the stuff. We can do print first record keys. Why is that a problem? Seek record object has no attribute keys. Print first record. First record dot annotation dot keys. What? Annotations keys. Jeez Louise. Okay, so this just gives us the actual keys for pulling each one up. So previously the annotations gives us the keys and then the answers, supposedly, or the response for them. This just gives us the actual keys themselves. If you don't care about what the answers are, but just to know what variables are in there. So we can also print first record annotations values, which will give us the other half of it, right? So, dot values. So as you can see, this just gives us the answers. So it's keys and values. So in general, these annotation values are strings or lists of strings. So they're not, even if they're a number like this, it's still a string, it's not an integer. What else can we do? Let's do print first record annotations, and let's do source, not sources, source. So this tells us the organism, and we can also do print first record annotations organism. Not organisms. Organism. There we go. And it's the same thing. So in this case, because we're doing this, let's see what this even is. I'm not a bacteria person. Oh, this is a plant. Oh, orchid, duh. Pelican orchid. So usually, I was thinking of the last sequence that we worked with. Usually, the source in most files will give you the common name. So usually this would say pelican orchid. And then, so if it's human, this would say human, and this would say Homo sapiens, right? But in this case, if there isn't a very common name, then it won't be included. So something like Arabidopsis, right? Arabidopsis, what I generally work with, Arabidopsis thaliana, everyone calls it Arabidopsis. It has another common name called Thalcress, but nobody really calls it that. So it might appear as Arabidopsis in most. But something like Mus muscalis, or mouse, or Homo sapiens, or things like that, Drosophila melanogaster, which is the fruit fly, then the source and organism might be different. Okay. Now, how about we do all species equals, and thus we'll create an empty vector. And then for seek record in seek io.parse, and then we'll do ls orchid gbk, and then we'll say genbank. Okay, so we're creating this loop. We're saying for each record that's in this genbank gbk file, let's see where I am. All species dot append seek record annotations, and then we'll append the organism name. Okay, so what this says is, all right, for each record in this parsed genbank file, append the annotation organism. So if you run this, do I type parse more or record? I don't know. Now we can print all species. Oh my gosh, today is just not a typing night for me. And so now we have a list of all 94 species that are in this document that we're comparing. So if you're like, oh, I wonder what species are in here. What was that, 2 lines of code? 2 lines of code. And you can see, pull out all the species instead of going and copying and paste everything. Okay, so now let's do all species equals seek record annotations organism, and then for seek record in seek io.parse ls orchid.gbk not comma genbank. So what happens if we run this? We're doing this slightly different. So now if we print all species, we now get this. This is so this is a list annotation. So it looks exactly the same, but if you've taken like my R class and things like that, or are familiar, sometimes the type of object is important, and so this just creates, it's just a different way of another reinventing the wheel, I suppose. Okay, let's see. I keep losing my spot. I need like a better text or a note editor thing because, or I need glasses. I think I do need glasses. It's part of getting old. Okay. Hold on. Where are we at here? Okay, perfect. Okay. Sorry for the delay. All right, so let's do all species equals, and we're going to create an empty vector again. For seek record in seek io.parse. Now we're going to do ls orchid FASTA. Oh my gosh. So we're doing it from a FASTA file and not a GenBank file. All right. So what happens if we run this? Of course we get an error. Oh, FASTA.text. Remember, I forgot to change that. Okay. There we go. So now if we print all species, you can see that we, from this, because the FASTA file, let's pull up the FASTA file real quick, it will still pull it up, but because it is annotated differently in the FASTA file, we get the genus being abbreviated and then the species name, as opposed to up here where we have the genus and the species. But you can pull from FASTA files in the same way that you can do that, or as you can with the GenBank files. So you can also do this the same way. I'm not going to redo it, but you can also do this for the list comprehension as well to make it a list. But we'll skip that. We'll have the same results. So let's do. Let's alter some data. So record iterator equals seek io.parse ls orchid.fasta. Jeez. FASTA. My goodness. I think I'm getting tired. All right, so now let's do. Let's run this first. Oh, FASTA.text. All right, I'm just going to rename this right now. So if you've been following my code exactly, then you might want to rename it too. But otherwise, put text in there. Okay. All right. First record equals next record iterator. So just remember, pulls the first one. First record .id, so we can pull this to see what our ID is. We get the. No, wait. Why is that? Wait a minute. Okay. Well, regardless of that. Oh, that's why first record. Huh. There we go. Okay, so not record. All right, so we get the first line of this file, right? Or at least part of it. That gives us the ID up to here, up to the name. And what if we wanted to rename this? So we could do first record ID equals new ID. And now if we run first record.id, then we've changed the name of it. So notice, if we look at our original file, it doesn't change it here. It doesn't write it in because everything's stored in the memory of our virtual machine or our computer in that case, right? So it's not a finished product yet. So we'll get to exporting the data or saving our data. But at this point, it's just in the memory that we've changed it. And when you save it, then it's not a problem, but it's finalized. And how about, let's do record, what's our first record? Okay, so we're gonna go work on the first record again. But let's add record description equals first record ID and a space and desired new description. So what happens when we do this? So now if we print the first record, oh wait, before we press for, record format FASTA, and then the first 200, or through, first 200. All right, so we're just gonna look at the first 200 characters. Maybe we put another parenthesis in there. Okay, so as you can see here, what we've done is we've changed the name, but then we've also added a description. So this is good if you do rename something, you can write why you renamed it, because they kind of have intuitive names. Well, not necessarily intuitive, but standardized names to begin with. So this is like, okay, let's change this. Let's do this. Mutations induced randomly. So say I went through and I said, okay, randomly throughout the sequence introduce 5 mutations because I'm doing some experiment with mutation accumulation or some simulation like that. So I could do that, and then when I pull it up, mutations induced randomly. So I know that when I pull it up, which one this is. So it's a nice way to be able to do that. 18 minutes already? Man, I can talk. All right, so we're gonna stop there. So my intimate camera is broken again. What's going on? What a bummer. All right, this one's gonna have another outro this way. So that is generally how we kind of pull the descriptions and things like that. We'll continue on with manipulating. You can see there's kind of overlap in some of these functions and things like that between whether you're working with a single FASTA file or you're working with these seq record files, things of that nature, or these GenBank files. There's a lot of overlap, but you do use all these different files if you do molecular biology or anything with genetics and things, bioinformatics, genomics, etc. ad nauseum. So you do use all these different files. So it's nice to know kind of the different ways that we go through them. And if you get one, then it's pretty easy to pick up the other ones. So thanks for watching. If you're watching YouTube, hit like, hit subscribe. I appreciate you guys that aren't forced to watch me watching, my students being forced to watch me, they elected to take the class, it's not required. So thanks a lot, and I'll catch you in the next video.